durante este tiempo fue forjando una gran trayectoria en los estudios de filogenias. Para aquellos que están un poco familiarizados con la evolución, pues realmente que muchos de los estudios evolutivos se basan en, utilizan como base árboles filogenéticos. Sin embargo, pues realizar estos árboles filogenéticos y afinarlos es toda una rama de investigación, es infinitamente complicado, y las ramificaciones de las distintas metodologías pues tienen mucho impacto sobre las conclusiones. Mario realizó un estudio muy bonito hace ya algunos años en donde pues presentó una filogenia de mamíferos utilizando metodologías muy novedosas, y pues fue uno de los primeros estudios que planteó que la radiación de mamíferos que tenemos y que vemos hoy en día, pues tal vez estuvo bastante ligada con el evento de extinción masiva que ocurrió hace 66 millones de años. Entonces es un área muy importante para todos los que tengan algún interés en áreas evolutivas. Es un gran placer tenerlo aquí desde el año 2015. Es académico de la Universidad de Meade, donde lideré ahora un grupo de investigación abocado a estos temas. Entonces, Mario, un gusto verte de nuevo y un gusto que hayas podido aceptar nuestra invitación. I'm gonna, for those who are listening or only speak English, many thanks for joining us. Our speaker today is Dr. Mario dos Reis from Queen Mary University, London. He moved to the UK in 2000 and studied his PhD in Birkbeck College, London, followed by research positions in both the Francis Crick Institute and UCL. And from 2015, he has been an academic at Queen Mary University, London. He is an absolute expert in many topics related to sequence evolution and phylogenetics. So it's an absolute pleasure to have him here today with us. So many thanks.
Okay. Okay, can you see my screen?
Yes.
So, okay. Bueno, muchas gracias por tenerme hoy, por haberme invitado. La charla, bueno, es una charla que he dado anteriormente, que la he ido modificando y siempre la he dado en inglés, así que pensé que tal vez sería mejor seguir dándolo en inglés, así no me confundo con los términos bayesianos de los que voy a hablar. Sin embargo, al final de la charla, si quieren pueden hacer preguntas en inglés y en español, no hay ningún problema, pero la charla la voy a dar en inglés. Okay, so thank you very much for having me here today. So I'm going to be talking about the research that I have been doing for the past 10 years, basically on using Bayesian statistics to analyze fossil genomes and their uncertainties in the molecular clock. So if you look, if we collect molecular sequence data for a set of species, for example, we have 8 species here, A, B, C, D, F, and G, and H, which could be any organism that you can think of. We collect some DNA from those species and we sequence that. We can align the DNA sequences, and then we can look at the patterns of similarities and differences along those sequences. And based on those patterns, we can work out the evolutionary tree depicting the relationship between these species. This is relatively straightforward to do. A molecular sequence data is very, very informative about these kind of relationships. But a slightly harder problem, and this is the problem that I'm interested in, is once you have built an evolutionary tree like this, how can you convert this splitting event here to divergence times in an absolute time scale? okay, in millions of years ago. And this is what I'm going to focus on today. Interestingly, we can actually do this because of a property of molecular sequences that we now call the molecular clock hypothesis. Okay, so this was proposed initially by Zuckerkamp and Pauling back in 1965. The idea is, or what they noticed when they analyzed the first protein sequences that were obtained at the time, between human, chimpanzee, gorilla, and other mammals, when they lined up these protein sequences for the globins, they look at the patterns of amino acid mutations between the different species, they noticed that the number of mutations between the species correlated very well with divergence times from the fossil record. In other words, more distantly related species, according to the fossil record, show more amino acid mutations between the globin genes. So if you will kind of look at a plot of the number, for example, of DNA substitutions in a molecular alignment and the geological time of divergence between species, you'll see a linear relationship like that. When this is showing you is that the rate of evolution at the molecular level is approximately constant in time. So we now know it's not perfectly constant, so we'll talk a bit about that. But this rate constancy, which we call the molecular clock, is what allows us to convert evolutionary trees from molecular sequences into evolutionary trees that have been calibrated to geological time. So I'm going to give you an example of, here it is. I'm going to give you an example of evolutionary tree. So this is a tree built using the mitochondrial genomes of human, Neanderthal, chimpanzee, and gorilla. And the numbers in the branches are the estimated number of substitutions or DNA substitutions in the genomes of these species. What would like you guys to see here is that if we look at the distance between the human chimp ancestor, so we have this node here called HC. If you trace, you go along the branches of the phylogeny until you reach human, you will see that the distance, the molecular distance between the human chimp ancestor and human is 7.57%. If you repeat that procedure by going from the human chimp ancestor to chimpanzee, you get a distance of 7.27%. So you can see that the molecular distances on the human side of the tree and the chimpanzee side of the tree are very, very similar. In fact, if you calculate the ratio between these two numbers, between 7.57 over 7.25, you get a ratio of 1.04. So this is basically what we call the molecular clock, all right? So basically, the number of mutations that have accumulated independently in these two parts of the tree are very, very similar, all right? So this rate constancy is what is going to allow us, and that's what I'm going to show you later, is that it's going to allow us to convert a tree like this into a tree that has been calibrated to geological time. Now, as a counterexample, I'm going to show you another tree. If these are the same species as before, but the difference here is that the length of the branches in the evolutionary tree have been calculated using morphology. Okay, so in the same way that we can generate a DNA alignment and count the number of mutations, we can actually take measurements, in this case of the cranium of these four species, and we can line these measurements up and we can see how divergent they are when you compare one species with another one. And that gives you a measure of the morphological evolution of the species. And you can see that this tree straight away looks very, very, very different to the previous tree, all right? If you look at the distance between the human chimp ancestor to modern human, the morphological distance is, for the cranium is 56.4 morphological units, all right? So these are the units of diffusion in morphological space. If you repeat that calculation from the human chimp ancestor to chimpanzee, you get a distance of 6.96. And the ratio of the two measures is 8.10. So what this is telling you is that morphological evolution in the human side of the tree has been 8 times as fast as evolution in the chimpanzee side. And that's not very surprising. I mean, if you compare the human cranium to other apes, we are the ones who have shown all these modifications that happen actually relatively quickly. So this is an example of non-clock-like evolution, right? So here, natural selection and other processes have accelerated evolution on the human tree. So if we go back to our example, our molecular example here, so we have this tree, which as explained to you is clock-like, and you can see that the tips of the tree kind of line up. So how can we convert this to evolutionary tree with geological types of speciation? Well, we can do that if we look at the fossil record, okay? So for example, a whole bunch of paleontologists think that the oldest fossil in human lineage is Sahelanthropus, right? So we can date to around 6.5 million years ago. So that will be a little branch here that sticks out in the human side of the tree. We don't know exactly the length of this branch or where it goes exactly because we do not have molecular data for this species, okay? So we know it belongs here thanks to morphology, but we don't know the distance. But we can use that information to try to calibrate the tree, all right? We know that the human chimp ancestor cannot be younger than these 6.5 million years, all right? So that gives a minimum bound to the age of the human chimp ancestor in the tree. But that's only a minimum age. You know, I had a chat with some paleontologist friends of mine, Phil Donahue and Rob Asher, and they think, and they've written papers about this, they think that it's unlikely that the human chimp ancestor is much older than 10 millions of years ago, right? And this is based on their analysis of the fossil record and patterns of fossil preservation. So that gives us a putative time range for the human chimp ancestor of between 6.5 to 10 million years ago. So now that we have this prior estimate for the age of the human chimp ancestor, we can use that information to convert the other node in the tree to a geological time of divergence. And we can do that very easily, simply because of the molecular clock hypothesis, all right? So we just look at the distance between the human chimp ancestor to human, which is 0.0757, and we can divide, so we get the distance between human and the human Neanderthal ancestor here. This is the other node that is labeled HA, so that's the human Neanderthal ancestor. So if you divide the two distances and you multiply by the 6.5 million years of divergence, that gives you an estimate, a minimum age estimate for the human Neanderthal ancestor of 0.62 million years ago. We can repeat that exercise using the 10 million year figure, and that gives you a full range, okay? So it gives you an estimate of 0.62 to 0.95 million years for the human Neanderthal ancestor. So this is how we can convert molecular distances into geological times of divergence for species. But this methodology that I use right now, which was very popular in the 1980s and 1990s, is what we call the naive molecular dating, all right? And the reason for that is because this procedure doesn't take into account all the uncertainties that are present in this type of data, okay? So for example, it would have been nicer to use some statistical distribution, for example, to represent the uncertainty of the human chimp ancestor, right? So we could have a statistical distribution represented here by this red line, okay? The fossil age will be the vertical line, but fossils could be wrong. So we could have a small probability, for example, that the fossil is wrong, and that's why the red line goes over. So that can be modeled. Not everybody, for example, believes that Sahelanthropus should go in the human side of the tree. If that's true, that would be the wrong age. At the same time, the previous example, we just use an arbitrary bound of 10 million years, but some people think it's better to have a statistical distribution that has a long tail. So we can do things like that, right? That would be more sophisticated. But also other bits that we need to pay attention to. For example, these branch lengths here have errors, right? So we ignore that. When we look at the number of mutations that have accumulated along a lineage in the tree, that's actually an estimate, right? Because you can have multiple substitutions, and we don't know what the true number is. And there's an error on that. And we can represent that error on the branch lengths, again, using a statistical distribution. And finally, we now know that there is no perfect molecular clock. We know that the evolution of the evolutionary rate accelerates in certain lineages and slows down in other lineages. So it's not right, and people don't consider it a good approach anymore, to use the same rate for the whole tree. So instead, we again use a statistical distribution where we try to model how the molecular rate varies on different branches of the tree. So we have all these sources of uncertainty. The question is, how do we put all these things together into one single integrated analysis? So the breakthrough for this is to use Bayesian statistics, all right? And the Bayesian method for molecular clock dating was first proposed by Jeff Thorne and colleagues in a series of papers between 1998 and 2002. The Bayesian method is very well suited for clock dating because it is very elegant, it's very straightforward. So you simply take these statistical distributions that represent your uncertainty and you multiply them together. So all the fossil uncertainty and the uncertainty on the ages of the nodes in the evolutionary tree will be represented by this FT statistical distribution, which is represented in blue here, all right? We will have another distribution for the evolutionary rate along the branches of the tree, which is represented here in green. And the uncertainty due to the molecular data, okay, so the branch length, due to the branch length, is given by the likelihood function here, which is represented in red. And literally, all you have to do is multiply these three statistical distributions together, and you get your estimates, which is known as the posterior distribution, shown here in orange. So it's very elegant. I have another symbol here, which is theta, the Greek letter. That simply, it means we'll have some model and there will be other parameters in the model, like speciation rates and things like that. So the Bayesian method is very flexible, so you can add more things. So I'll show you an example of how all this works. So this is an example of an application of the Bayesian method to estimate the divergence time between two species. So here we only have two parameters to estimate, will be the divergence time between the two species, which will be the age of the root of the evolutionary tree. And we will have to estimate the molecular rate for the two species. So the first square here is the prior, all right? Usually we tend to have good information or relatively good information from the fossil record. And that means we can build a prior distribution on the age of this node. You can see that on the x-axis, okay, which represents this uncertainty on the age of the root, the distribution is relatively narrow. The y-axis on the first square represents our prior uncertainty on the molecular rate. Usually, a priori, we might not know what the molecular rate is, so we might choose a prior uncertainty which is more diffuse. So you can see there is a much longer tail on the y-axis. The square on the right shows the likelihood, all right? So this is the probability of seeing a particular molecular alignment given the times and the rates. The molecular distance is the product of the time and the rate, right? So if we have a divergence time of 1 million years and a molecular rate of 0.5, we will get a molecular distance of 0.5. So the issue here, and this is very, very important in molecular clock dating studies, is that molecular sequences only have information about the number of mutations separating the two species, but the molecular sequences themselves do not have information about the time or the rate separately. Okay, so in this particular example, on average, we see 0.5 differences for each nucleotide site in the alignment. And this curve here, which looks like an L-shaped curve on the right side, is the curve which is compatible with all combination of the rate and the divergence time that give you a molecular distance of 0.5, right? So for example, if the age of the root was 0.5 and the molecular rate was 1, you'll get a distance of 0.5. So that would be compatible with your data. But on the other hand, if you were to assume that the age of the root is equal to 1, and the molecular rate was equal to 0.5, that also gives you a molecular distance of 0.5. So, sorry, all the points on this blue line here are the points that are compatible with the estimate. So this is our prior and this is our likelihood. In the Bayesian method, all we have to do is multiply them together. All right, so when you multiply them together, you get your estimate, and you can see how things change now. So together with the prior and the information and data, we can now pinpoint what the estimate is. In this case, because this is a very simple example, we only had 2 species, we already had a good idea what the divergence time of that node was. Okay, so here, the most dramatic change is in the estimate of the molecular rate. So you can see what happens here. Okay, so this little roundy cloud here that you see on the right side of the slide is a posterior estimate of the divergence time and the evolutionary rate of these 2 species. Now, and here comes a challenge in molecular clock dating analysis, because the data, the molecular data, only gives you information of the total distance. If you change the prior, you will change your estimates, right? So for example, a different scientist that thinks you should have a different fossil to calibrate U3 and a different prior on the rate will generate a different estimate, okay? And this is why we say that because of this, Bayesian molecular clock dating is an unconventional statistical problem. So you don't see this in other type of Bayesian problems. This happens here because of the nature of how the molecular data is constrained of the information that you can get out of it. So we say that the divergence times of the species and the evolutionary rates are unidentifiable in the likelihood function. So that has very important consequences. So I'm going to show you some results. So this is a paper which I did with Heng Yang in 2013, where we basically look at how Bayesian estimates of divergence times are affected as you collect more and more molecular data. So the top panel here shows you the prior, the likelihood, and the posterior, again, for the analysis of 2 species. All right, so we have a very diffuse prior here, panel A, top, and then we have the likelihood function. In this case, we were analyzing a molecular alignment of 100 sites that show 55 differences. So again, we multiply the prior and the likelihood together, and we have a posterior estimate here in panel C. The bottom panel is similar to the top one, but the difference is that we're using a much longer alignment. So we analyze in the bottom panel 1,000 sites instead of 100. And it has the same estimate for the distance, right? We see 55 differences, which also gives you a distance of about 0.55. So when we multiply the 2 together, we get the posterior distribution of the divergence times. But what happens when you compare the 2, what you see is the distribution becomes very narrow, okay? And this is because the uncertainty on the branch length has been reduced because we have collected more molecular data. However, the uncertainty on the estimates of the rate and the divergence time has not really changed, okay? So this surface here actually looks like a mountain, okay? And the estimates of the time and the rate are actually the views of these mountains from the side. If you look at the mountain from the south, that'll give you your estimate of the time. If you look at this mountain from the west, that will give you the estimate from the rate. And I'm going to show you how that looks like, all right? So this is basically in a 3D depiction of this posterior distribution, all right? So this is how it looks like. And so when you actually look at the marginal distributions, which is at the times, and the rate separated, you get something that looks like that, okay? So we get here the posterior distribution of the time and the posterior distribution of the rate. So you have the prior here, which is the dotted line. That's our prior belief before we add data. And you can see what happens as we add more and more data. So we analyze an alignment with 10 sites, 100 sites, 1,000 sites. And we can calculate as well analytically how it looks like when we have an infinite number of sites. And you can see that when we have 1,000 sites, the posterior distribution is very, very similar to that you obtain with 100 sites. So there's almost no difference, all right? So in other words, so there is a limiting distribution for the uncertainty of the divergence times and molecular rate estimates in these kinds of analysis. So this is very challenging, because what that tells us is that setting up your calibrations from the fossil record before you do the analysis is extremely important, because the analysis is extremely sensitive to this. Another challenge that we have when we use molecular clock dating is the analysis of large data sets. We now have complete genomes, and people are interested in, wind economic and alignment using 30 mammal species, for example. We have 30 genomes, and we align all that. But there is a challenge with that. And this is that the more data we have, the slower the analysis is, right? So on the one hand, ideally, we would like to look at the behavior of time estimates when we have very, very large phylogenomic data sets. But we cannot do this because we have this little normalization constant in the Bayesian equation here, K, that needs to be calculated. So I didn't talk about that, all right? But when we talk about probabilities, so when we do a Bayesian analysis and we calculate the posterior distribution, we can then say things like the probability that the divergence time of this species is between this and this age is 10% or 20% or 90%, all right? And probability is the maximum probability you can have is 100%. So to do that, we need to rescale the posterior distribution so that the total integral of the posterior distribution is 100%, which is the maximum probability. And you do that by calculating this little constant K here, which we call the margin, the normalization constant of the posterior distribution. And this little constant is actually a very complex integral, which is very, very rarely available analytically. So it cannot be calculated. And because of this constant, the Bayesian method in general was very limited for a very long time, right? So the solution to the algorithm that we use to overcome this problem of the normalization constant is known as the MCMC algorithm, which uses a stochastic simulation to obtain samples from this posterior distribution. And I'm going to show you quickly how that works, okay? So suppose you have an evolutionary tree of 3 species that we have here, and we want to obtain the posterior distribution for the divergence times and the evolutionary rates in the tree. So in this example, because we have 3 species, we have 2 divergence times, okay? We have the ages of these 2 speciation events. We have 4 branches in the tree, so we will have to estimate 4 evolutionary rates. So we'll have in total 6 parameters. And what we do with the MCMC method, we just choose a starting configuration for these parameters, all right? So we start with a random tree that has some random times and some random rates. Then what we do is the computer performs a random perturbation to one of those parameters. So in this case, we do a perturbation to one of the nodes, indicated here by the red arrow. So we made that node older, so we push it down compared to the previous one. So we have done down our perturbation. So what we do is we now calculate the posterior for the perturbed data over the posterior from the initial data. And this is this equation that we have here. So the perturbation is indicated with the asterisk. Now, what happened is that because we're dividing the 2 posterior distributions, the normalization constant cancels out, all right? So that's the great advantage of the MCMC algorithm. So when we do that, we don't need to know the value of K, all right? So we calculate that. 2 things can happen. If the probability of the new tree is higher, in other words, if your perturbation leads to a parameter value which is more likely than the previous one, then this ratio will be larger than 1, and you will accept that perturbation straight away, okay? If the probability of the perturbation is lower than the previous step, you still might accept that perturbation, but you will accept it with a probability equal to the ratio. So if the new tree is half as likely as the previous one, then you will accept that new tree with a probability of 50%. all right? So if you accept the perturbation, you move on. If you reject the perturbation, you just go to the previous step and you do a new perturbation. So in that way, you can accept or reject perturbations, and you repeat the procedure over and over again for all the parameters in your model. So we will do that here for the 2 divergence times in the tree, and we will do that for the evolutionary rates. Okay, you do that thousands of times, millions of times. When you finish, you just get a table. In this case, you'll have 6 columns, which is the 2 speciation times and the 4 evolutionary rates. And the rows will be the sample that you obtain in the random perturbation. And then you simply look at that. So for example, if you build a histogram from your sample, that histogram, which you call the MCMC histogram, is an approximation to the posterior distribution for that parameter. So that's how we overcome the problem of the normalization constant. So a typical Bayesian analysis for molecular clock dating can take a few hours or even a few days or weeks, right? Because you have to do this simulation algorithm that has to be repeated over and over again. So this then obviously becomes very challenging, because when you calculate the Bayesian equation, so it is straightforward to calculate the prior and the likelihood. However, calculating the likelihood is slow because the time that it takes to calculate the likelihood depends on the length of the alignment. So the more molecular data you have, the longer it takes to calculate the likelihood. Because of these new genomic data sets that we have now, people became very interested in using these large data sets for molecular clock dating. But it turns out that calculating the likelihood for these very large alignments is so slow that when you apply the MCMC algorithms to these data sets, it will literally take years to run the algorithm. So it was not feasible. Okay. So again, Jeff Thorne proposed an approximation for the likelihood so that the MCMC will be faster. And this is just a mathematical trick. It uses something that is known as the Taylor method. We can just approximate the likelihood using Taylor expansion. So the advantage of this method is that it then reduces computation times dramatically from several months to days. And it makes analysis of these very, very large scale data sets feasible. Okay, so I mean, the first method that Jeff proposed was in 1998, but his program was quite slow. In 2011, myself and Heng, we re-implemented Jeff's approximate method into MCMC tree. So we did a version that was faster, but we also look at ways to improve the approximation. So if we look, I'm going to show you in the next slide. So this plot here is the likelihood function of the molecular distance. So the molecular distance is in the x-axis here. And the vertical axis is the log likelihood. The solid line is the true likelihood. So this is the likelihood that you will obtain if you calculate it exactly. Then we have the original method developed by Jeff Thorne, which is the NT approximation, which is the normal Taylor approximation. So this approximation is symmetrical. All right, so you can see that here, which is the large dashed line. It's symmetrical, but if you look at the true likelihood function, you can see that the true likelihood is actually asymmetrical. So the original method by Jeff will overestimate the likelihood for small branch lengths, and will underestimate the likelihood for long branches. So we look at further mathematical tricks. So we use what is called transformations of the branch lengths, square root transformation, logarithmic transformation, arc sine transformation, to try to improve the approximation. And you can see that transformations do improve the approximation. In particularly, this arc sine transformation leads to an approximation that is almost indistinguishable to the exact likelihood. So we implemented that in MCMC tree program, and we also did other tricks that made the program much faster. So once we did that, so the next question was, okay, so now we have a reliable method to approximate likelihood, and that can now analyze these very, very large genomic alignments. So what we did next was to use our new method to try to understand the diversification of the mammals. So this was the first Bayesian method or using complete genomes on mammals. Mammals have been very studying the diversification of mammals has been very controversial. I mean, I'm not the first one. So Araxi said I was the first one. I'm not the first one. This is a very old problem. So this tree here, so this is a tree of mammals from a paper that was published in Nature by Luo. What I like to show you in this tree is that this shows modern mammals and extinct mammals. So the modern mammals, the ones that are alive today, are only three groups, which are the monotremes, which are the ornithorhynchus and the echidnas, the marsupials, right, and the placental mammals. So you have the three groups here. But you can see that there's actually a very large diversity of mammals that existed before and that became extinct. And the majority of those mammals became extinct 66 million years ago in the famous Kpg extinction. All right, so the Kpg extinction was when we have this large asteroid, comet, or this extraterrestrial object that actually hit Earth. All right, and it actually hit in the Yucatan Peninsula, and we now know because we found the crater, and the crater dates exactly to 66 million years ago, which is the time in the fossil record where we see all these species of birds, mammals, lizards, and many other species throughout, not just dinosaurs, but 70%, 76% of all the species that were alive at the time became extinct. All right. So three mammals, as I said, three mammal groups survived. When paleontologists look at the fossil record, traditionally before we had any molecular data, the pattern that they saw was we have a group of mammals living with dinosaurs before the Paleogene. We have the big extinction, and then three groups of mammals survive, and then these groups of mammals diversify after the extinction. So that was the view from the fossil record. And we have some examples here. So this is some papers by Alroy and Archibald. So both papers show number of mammal species and number of mammal genera and mammal orders with time. And you can see in both cases, the number of placental mammals increases sharply after the extinction event shown by the red line. Okay, so this was the status of paleontological analysis up to the late 90s. Well, of course, as soon as we started collecting molecular data, people started to use what I call the naive molecular clock dating to try to estimate divergence times of mammals. And we have this very famous paper that came out in Nature by Kumar and Hedges in 1998. So they analyzed, if I remember correctly, over 800 genes, and they analyzed them separately. And they use a calibration of the divergence time between birds and mammals of 310 million years ago. But the issue, though, is that if you look here, you can see the extinction event in this red triangle here on the left. And you can see all these modern placental groups estimated to have originated deep in the Cretaceous. So this was in complete contradiction to the fossil record, because we don't have fossils of horses or fossils of rodents or bats or monkeys at that time. All those fossils appear after extinction. So this paper was incredibly controversial. And this controversy had already started, but this really made the whole thing kick off. And it was the paleontologists against these new molecular biologists doing these divergence time studies. So the thing got very heated up because it was very puzzling to have this molecular data giving such ancient ages of diversification when the fossil record didn't show that. Many papers were published after this work by Kumar. So this is a paper in PNAS. So this paper actually, this is the first Bayesian paper on a small data set. They used Jeff Thorne method, and they actually found divergence time for placental mammals that were much more closer to what you expect in the fossil record. So this paper in PNAS reconciled a little bit those dates. But then we have other papers. So we had another paper that came out in Nature by Beninda Emmons in 2007. This paper, again, analyzed a very, very large data set of almost 5,000 mammals. And again, they got these very, very ancient estimates for placental mammals deep in the Cretaceous, again kicking off the controversy. And again, there was another paper in 2011 by by Meredith et al., kind of a middle way between the other two. So this became incredibly controversial. So up to this point, and I can show you here why this was so bad. So this is looking at the estimates of divergence times from benign diamonds. So for example, benign diamonds found bats, which are the Chiroptera, originating 4.9 million years ago. But the oldest recognized crown bat fossil, Icaronicteris, is around 48.6 million years. So that's a gap of 26 million years between the age of the oldest known bat fossil and what benign diamonds found to be the estimate for this group. The same for primates. Again, they give an estimate of almost 90 million years ago for the origination of primates, but the oldest crown primate fossils are around 55, 66 million years. For rodents, again, they get 85 million years estimate. The oldest fossils are only around 55. So we have this pattern of fossils being present after the extinction. We have these very old estimates. In total, benign diamonds found 9 out of 18 mammal orders originating in the Cretaceous. So that's the 2007 paper. So this again reignited this big controversy. But again, this paper was done using some of these more what I call the naive molecular clock dating. So we wanted to look at this again, all right, and we wanted to distinguish between these two scenarios of mammal diversification, all right. So we have a model. I mean, obviously, the ancestor of all placentals must have survived extinction. So one model says we have ancestor of placentals in the Cretaceous with the dinosaurs. After the extinction, you had a diversification, and there was a morphological diversification, and then the modern groups appear. So this model here on the left is what we call the short fuse model, and is the one that is compatible with the fossil record. And the model on the right is the model that people like benign diamonds and Coumaran hedges will favor, which is you have divergence of these modern groups here shown in orange occurring deep in the Cretaceous with dinosaurs. And this is the model that has been very, very controversial when compared to the fossil record. So we had a new method, and we wanted to look at it, and we wanted to overcome some of the problems, particularly the benign diamonds and the Coumaran hedges papers. Both papers ignore uncertainty in branch length in the tree, so they just estimate the distances, and that's what they use. The Coumaran hedges papers use a strict molecular clock. I mean, they do some corrections, and if some genes have too much variation, they remove them. But benign diamonds does has a kind of way to correct for it, but it's not very good. And both papers completely ignore uncertainties in the fossil record, so they just use point calibrations for the nodes of the tree. So so after we published a paper in 2011, we then went on and we collected all the available genomic data for mammals at the time, which was 36 species, with complete nuclear genomes. We also collected all mitochondrial genomes available at the time, which was 274. So in total, we generated an alignment of 14,000 orthologs, which equated to almost 21 million aligned base pairs. Obviously, our objective was to use the Bayesian method and try to put all these sources, to try to account for all these sources of uncertainty that I had described before. We use 26 fossil calibrations to calibrate our tree, or obviously to construct the trend prior. And these calibrations were actually prepared by Phil Donoghue and Rob Asher. So we got two paleontologists that are, some of the pioneers on constructing these calibrations, and we asked them to prepare those calibrations. And the idea was, when I started this work, I didn't have an opinion one way or another, right? I just wanted to see what was going to happen when we use the Bayesian method on complete genomes, with these fossils. So can we say anything? Can genomic data tell us anything about this pattern of diversification of mammals with respect to the extinction? So I'm going to show you, this is another beauty of the Bayesian method. When we calculate, before we put the data, we can actually calculate the prior only. So the left panel here is the evolutionary tree of mammals calculated from the fossil calibrations only, without any molecular data, right? So we haven't added any molecular data here. So the fossil calibrations that Phil Donaghy and Rob Asher prepared were actually very diffused. The uncertainty is shown here by these blue horizontal bars, all right? You have two red circles here. So that represents the ancestor of all the placentals and the ancestor of that ancestor, right? So that shows the branch length, okay? So this is the placental branch length. You can see that the tree is very diffuse, and you can see for a lot of mammal groups, the blue bars actually overlap the extinction event. So if you look at the prior only, the prior that we use allows for modern groups diversifying after the extinction or before, right? So this is what the fossil data on its own, which was analyzed objectively by Rob and Phil, did. After we add the molecular data, this is what happens. So we now know, because molecular data is very, very informative about the molecular distances. So we now know that this ancestral branch to all placentals is actually very long. We know that because it has a lot of mutations that have accumulated. Again, remember that this is a method that relaxes the molecular clock on the tree. So each branch here has its own evolutionary rate, which is estimated from the data. And what happens is that because this branch gets very long, it actually, pushes the two sides of the tree. And what we get here is that much more recent divergence for the modern groups. So if you actually look, I'm going to show you a summary of the data here. So this is the full tree with all the 274 species. You have the extinction event here. So you can see that there are very few lineages in the Cretaceous. And you can see most lineages actually appearing in the Paleogene. Okay, and here we have a summary for the mammal orders. Yeah. So we have here the main mammal orders. So obviously each order is important because they show a specific morphological adaptations, right? Like for example, all bats are in the order Chiroptera, all primates are in their own order, whales are in the Cetacean and so on. So what we got, when you look at the data summarized in this way, is that all the modern placental groups, except primates and xenarthrans, which is a group of South American mammals, which include the sloths and the armadillos, all of them originated after or diversified after the mass extinction. And when you look at primates and xenarthra, we got estimates that just predate the extinction event by very little. So our paper was, much more in agreement with the fossil record than the previous one. And because we had used all these very large genomic data, and also we had used so many fossil calibrations that had been prepared by, Rob and Phil, kind of the paper was very convincing. So it was well received by people. So we try to, reconcile that. Of course, our paper has not abated the controversy completely. I mean, there's lots of papers came out. There was a paper that came out in 2013 by Meade. So our paper was from 2012. So the following year, there was a paper was analyzing fossils only. Well, there's some molecular data here, but it's a very fossil skew analysis, and they went the other way. So in this paper, they say that all the placentas, including the ancestor of all placentals, postdates the Kpg extinction. So this is a very extreme view and a very extreme interpretation of the fossil record. We wrote a critique of that. There's another paper in CISBio. I mean, there's many, many more papers. This is just very few examples. And very recently, this is a very nice paper by Nathan Upham that just came in PLOS Biology. That just came in December last year. We look at very big tree. So Nathan's paper is more like also using the Bayesian method, and he gets the kind of results that we got. I don't think this is gonna end anytime soon. So Nathan's paper, for example, used about 5,000 species, but he used a very short alignment, all right? So he, because he has so many species, he could not use complete genomes. But this is just, I want to show you this. This is an example of how we can try to put all these things together to use Bayesian statistics to understand the molecular clock. I have, I'm not sure how many minutes do I have left. Maybe I can keep going. Okay, so yeah, I was going to show you very quickly another example that we did with the metazoans. This is the origin of all animals. I can very quickly go through it. All I wanted to show is that in the case of mammals, we have a very good fossil record, and we can get very good estimates. When you try to obtain divergence times, for example, for very ancient mammals that originate in the Cambrian, fossil record is very imprecise. The molecular data is much more uncertain. And when you look at that, it's very difficult to resolve. And this is an example that had a similar controversy of whether animals originated after the Cambrian or before. It's a very similar story to mammals. But when we did analysis, actually the result that we got was that we couldn't tell, because there's so much uncertainty so far back in time that we got analysis where animals could start deep in time or not. So, I mean, this is a paper that we published in Coordinan Biology. It's just, I wanted to show that as a counterexample of that, and how the molecular clock here changes so dramatically that you cannot really analyze it. So it was like the counterexample. And just briefly, because there is no time, what I want to show you is the future of the method, which is to use morphology. I didn't touch on that, but we have this. We've been using fossils in the previous analysis as just numbers, all right? But there's these new methods where we can actually integrate fossils as data, and we can use Bayesian statistics, and so we can generate these beautiful Procrustes alignments of fossil shapes, and we can integrate that in the Bayesian model, and now we will have a likelihood from the genomic data with a likelihood from the morphological data, and now we can estimate. So these models are very new. We have a paper on that, which I was not going to talk about that today, but I just wanted to show the future of the Bayesian method right now. So people are working on this discrete morphology, and I've been working on this continuous morphological characters. But this will be material for a different talk in the future. So, well, I would like to thank a lot of people, especially my collaborators, Zihen Yang at UCL, Phil Donahue, Greg Gonell, Max Telford, Rob Asher, Masami Hasegawa, and Jung Inoue, which is all these collaborators that work in these papers with me. Also, I would like to thank Araxi very much for having invited me today, and obviously all the organizing committee, which I don't have a space to name everybody, but the organizing committee and the Fundación UNAM, that it's very kind of them to have invited me today. And I think it's a great idea that you're organizing this. I think it's just fantastic. And this will be in YouTube and people can see that. And obviously I would also like to thank my PhD students, Sandra, Konstantinos, and Yutapon, which actually did a lot of the heavy lifting from some of these papers, particularly for the metazoan paper, of which I didn't talk about. Kostas and Yutapon did a lot of that. And Sandra has done all this work on morphology that we just published last year, and she's been excellent. And it's really been really nice. So thank you very much. So if anybody has any questions in English or Spanish, that's okay. I'll be happy to answer them.
So, muchísimas gracias, Mario. Thank you very much for that excellent talk. So we do have some time for some questions. The first one is, well, you only very briefly touched on the metazoan case and the controversy as to whether the radiation happened before or after the boundary in the Cambrian. And you say that actually your results are that we don't know. Do you think that can change? Because presumably a lot of the additional morphology data from the fossil record, perhaps to some extent, it's going to be limited going that far back. So
yes, yes, that's a good question. So I mean, the Cambrian example, I have the tree here, is been very puzzling. I mean, first, for a very long time, all animal fossils were in the Cambrian, and you have all the many things that look like modern groups, like arthropods and jellyfish and all these things. And there was nothing before that. We now do have fossils in the Ediacaran, which is the period before, but these fossils look very, very different to what we get after the Cambrian. So that's where the controversy is. So some people, the fact that we now do have animal fossils in the Ediacaran makes a lot of people think that many of these animals were obviously before, right? So these Ediacaran animals are clearly the ancestors of what we have now. It's just that they look so different that we don't know how we don't know if they are part of the cron group or stem, and there's a lot of controversy exactly surrounding that. The challenge for us here, I guess, is when you put all, first, we are not completely sure what the evolutionary tree should look like. So people are still fighting on whether this group is the basal to that one or not, like Xenoturbella and Deuterostomes and all these. There's these controversies going around right now. So those uncertainties are problematic. The other thing is that, as I explained before, the molecular clock is not constant in time. But what we see is that when you analyze closely related species, although the rates vary, the rates are similar. So when you look, for example, at apes, human, Neanderthal, and so on, we have our own rate, but it's very similar to each other. So, because there's very slight variation in the clock, there's no big problem. But in this animal tree, there's so much variation in the rates that it's almost like there is no molecular clock when you look at these deep phylogenies. So that's one of the reasons I complicated all that. I feel optimistic because here we only analyze about, if I remember correctly, I think we did about 200 proteins. But we now collecting much larger data sets. And now that we're getting complete genomes, on the one hand, by looking at the complete genomes, we are now starting to understand the relationship between these groups much better. So the uncertainties due to the tree topology are being resolved because we have more molecular data. That should also improve our estimates of the branch length. The question then is the problem of where do we put all these fossils? When Rob and Phil helped here in this paper, fossils were placed based on their interpretation where it should be. But if we use morphology, we can then let the computer program decide where the fossils can be. So there are some very sophisticated models that are being developed right now. The problem is that they're big computationally expensive, like the threshold model and these morphological landmark models. But I feel very optimistic because if we can include things like correlation between the characters, and if we can score many, many, many hundreds of characters in the species, I suspect this will start to have far more information about these more ancient events. It's difficult to predict what's going to happen because we're collecting this kind of data right now, and we hopefully in the next few years we're going to try to do this with more species. So it's hard to predict. I feel optimistic, but until we do this analysis with more data, we really don't know what's going to happen. But I do feel optimistic, as I said.
Yeah, and I think what you're saying is basically would reconcile this old, well, not that old, but like 20-year-old dichotomy between the fossil record and the morphological based trees versus the genomic or genetic sequence based trees. So basically you're saying fossils could be brought back into the equation as more than simple calibrating points.
Yes. And this is following from, this is actually follows ideas from other people like Fred Ronquist and all that, who started using discrete characters. I've been interested in using the continuous characters because I think they allow using correlations with characters much more easily.
Okay, so to what extent uncertainties on calibration and molecular rates become a problem not only of timing divergence between lineages, but start to become a problem in defining the actual topology of the tree. And for example, in groups where there is a very rapid radiation, as in birds, where you start not only having issues as to timing the divergence events, but actually you have problems in knowing what is the closest relative to what.
Yeah, so we had that same problem in this metazoan tree. So if I go back to this slide here, so we actually, when we look at the paper, because the approximate method that we developed can only date a single tree topology. So other Bayesian programs like BIST, for example, will rearrange the tree at the same time as the divergence times are being estimated. In MCMC tree, you need to fix the tree before you do the analysis. However, what you can do, if you think there are many possible topologies, you can analyze those independently. And in the case of the animals, and I was doing this with Max Telford, I said to Max, because he's been looking at these kind of things with uterostomes and all these ancient relationships a lot, I said, Max, we don't need to focus on a single tree. Look at all the controversies, what people are saying, and we can build all the candidate trees. And he came out with 162 possible rearrangements of the metazoan tree. And what we did, we dated them all. So some of these trees were incompatible with some fossil calibrations which were not used. Some fossil calibrations were compatible with all trees. But what we did all the analysis. And this plot here looks a bit small, but it's not. So the x-axis is the tree number. So we numbered the trees from 1 to 162.
Mario, ¿puedes compartir tu pantalla?
Oh, sorry, it's not there anymore. Oh, what happened? Sorry, let's do that again. So, can you see that now?
Yeah.
Yeah. So this from an image from the paper. So what we did, as I said, so Max Telford came up with 162 possible ways to rearrange the animal tree, and we dated them all. So this little plot here shows the 162 trees on the x-axis, and the y-axis are the divergence times. And each color represents a different node in the tree for a different group. And you can see how the ages, so the crown metazoan, which is the black lines, you can see how the ages shift depending on which of the rearrangements of the tree. So this is the ancestor of all animals, right? And even these ages change. So yeah, that's incredibly challenging. And we, until these relationships are not resolved, we really cannot know. But my advice for people doing this type of analysis is that you can do this. You can come up with all the topological hypotheses that you have, and you can date all these trees, and you can present the analysis in something like this. I will imagine that as we more and more genomes, we will be able to reconcile some of the topologies. Some of them will be eliminated. I mean, for example, for a long time, people were debating whether human and chimp went together in the tree or whether chimp should go with gorilla. That was very controversial, and there were so many papers on that. But nowadays, everybody agrees that it's human and chimp together. So that controversy has disappeared. I imagine that, in 10, 20 years, many of these controversies will be resolved as well. It's just a matter of as we collect more data. And I suspect one thing, people not looking at this, well, they're starting to look at it now, but the genome architecture. So, when you look at which genes are present and the position of the genes in chromosomes, that has a lot of information about relationships as well. So the method that we use now tend to ignore that, but these people are starting to look at this and incorporate that. So I think we'll be able to sort this. But yeah, it's very challenging.
So a question from Rafael Melendez is asking whether I think you partly answered this already. What is the resolution that you have in the molecular clock for hominins and apes in general? So how certain can we be on the timing of those events?
Well, that's improving. So we did a paper, actually, we published it in 2018. We will look again at all the primates using complete genomes for 10 species. And then we also combine that with molecular data for another almost 400 species. So primates are a good case because they have a very good fossil record. Well, it's not that they have a better fossil record, but, because the human connection is, there is clearly a bias on searching for primates fossils. So the collections of primate fossils are very extensive, and they cover the whole of the primate tree. I mean, if you look at the number of fossils just in the human lineage, it's just staggering. People still talk about the missing link, but it's not really missing link. We have now fossils almost going from present time to things. So there's less controversy now regarding primates, in my opinion. There's no much issues. I mean, the divergence between human and chimp, people used to think was around 4 million years and went to 6 million years. Then when Sahelanthropus was discovered, it's now been pushed to over 6 million to 7 million years. Some people are not totally convinced of that, but I think looking at all the majority of the papers that are coming out, I think between 7 to 10 million years, for example, is now tends to be people agreeing to that could be the divergence between human and chimp. But 7 to 10 million years, that's a 3 million year wide estimate, right? So it's still uncertain. So there's still a scope to improve this, but we are limited by the precision in the fossils.
Okay. So I have a question from Oscar García Miranda. He's asking, if we only had some limited funding, what would you spend your money on? Trying to find more fossils? Or trying to sequence more genomes?
Well, if they give the money to me, it will be just to develop better algorithms, because that's what I do. I've been working. So the problem that I have now is that, the method is very fast now for complete genomes. But if you add many species, it slows down to a crawl. So I work on methods that can use thousands of species to try to speed up. So that's how we use the money personally. No, but seriously, I think we have enough genomes right now. We have the Earth Biogenome and the Darwin Tree of Life projects, and they're already talking about producing, over 50,000 eukaryotic genomes in less than 10 years. And there's the 10,000 animals project. So I think we have already invested enough money on getting enough genomes. I don't think divergence times estimates will improve that much by having, it will have an effect, but I don't think that more genomes is going to help us. I do think we need more fossils, and I suspect paleontology is underfunded compared to sequencing. So I do think paleontologists need money to get more fossils. Yeah.
Okay. So there was a paper recently from Daniel Fields suggesting that there can be a lot of variation in molecular rates, even within a single lineage. So he was saying that, for example, variables such as organism size could have an effect. And in the case of mass extinctions, it could actually have a significant effect on timing radiations, since you would have a huge change in the average size of organisms used in time 1 and organisms present in time 2.
Yes. So there's been quite a few papers actually, people talking about, because there's a correlation between the body size, generation time. So things that are small tend to have shorter generation times, and they tend to have faster per year molecular rates, while large things that live longer, that have longer generation times, tend to have low things. One thing that we do notice, there are two models that you can use when you do these clock dating methods. You have what they call the independent rates and the autocorrelated rates. My opinion is that the independent rate models for things like mammals and vertebrates is completely wrong. Well, it's a model that cannot even be constructed using stochastic probability theory. Rates have to be autocorrelated because the rate of evolution of organisms depends on the ancestral rate. So if you analyze the data, for example, in the case of the mammals or the primates, using the independent rates or the autocorrelated rates, you get very different results. The independent rates tend to be very broad and give you things that are much older. The independent rates assume that the variance of the rate is the same whether you analyze in closely related species or distantly related species. And I think this is very problematic. I think this assumption, together with the uncertainty in the fossils, become very toxic, and the independent rates model just fills in everything, and you get very strange estimates. If you do autocorrelated rates, things become very different, because as you go back in time, you need to join these rates. And what you get is that the estimates of the rates in the autocorrelating model, the base of the tree, look different, all right? And I suspect that somehow this model is tracking this body size, generation time situation better. There's been people like Nicolas Lartillo. He wrote a paper where he explicitly was modeling correlation between the rate and body size. So it's a paper in MB to do that. And we don't have that in MCMC3, but this is something that we could explicitly do. So we could have, because we have the continuous character paper that I did with Sandra, we could combine things like body size as a continuous character that could be correlated to the rate, and you can estimate that in the Bayesian model. Yeah. Oops. Araxi is gone. Okay.
Thank you. A second, we lost a connection with Araxi. Doctor.
Sí, muy bien. Realmente, pues, muy interesante la conferencia. Mario, nos ha dado mucho gusto. Tenemos, de hecho, todavía varias preguntas, pero yo creo que podemos con gusto pasártelas y ver de responder. Hay incluso algunas sobre, con el último ejemplo de la explosión del Cámbrico, de pues, la capacidad que tiene el reloj molecular para tiempos extremadamente largos, es muy interesante. Y yo creo que esta combinación de reloj molecular y fósiles pues tiene un largo camino todavía por delante. Realmente sí. Ha sido muy interesante el escuchar tu conferencia. Ya regresa nuestra coordinadora, si quiera poder seguir.
No, ya terminamos con las preguntas. Podemos cerrar. Una disculpa, pero es que se me terminó la pila y no conecté rápido.
Muy bien, sí, bueno, pues estamos comentando nada más que pues el avance que se ha tenido en el reloj molecular es una de las herramientas, yo creo que ahora podemos aprovechar y tener para ir resolviendo cómo ha sido la evolución de los diferentes grupos y con un detalle que se antojaba muy difícil de lograr en el pasado. Entonces, pues es pues muy estimulante el escuchar la presentación de Mario. Y bueno, tiene la parte de las, estimar los tiempos de las divergencias de los diferentes grupos y las diferentes especies, y también la parte de las extinciones. Toda la discusión sobre la extinción que acaba con los dinosaurios, pues realmente es algo que ofrece también una enorme resolución para resolver buena parte de las controversias que tenemos. Entonces, pues agradecemos muchísimo a Mario, agradecemos a todos los que nos han estado acompañando en las diferentes plataformas de Fundación UNAM y del colegio y del consorcio de universidades por la ciencia. Les invitamos a que nos continúen acompañando. Este jueves tenemos la presentación de un documental que está también muy relacionado a la situación en la que estamos en los diferentes países sobre la parte de la depresión. Es el jueves a las 6 de la tarde por la plataforma de Fundación UNAM. Y el próximo martes tenemos la siguiente conferencia del ciclo de Universidades por la Ciencia a cargo de la doctora Catherine Peichel de Suiza, en donde vamos a seguir comentando sobre la genómica de adaptación. Tenemos muchas felicitaciones a Mario por la conferencia de diferentes lugares y saludos también para la continuación del ciclo en varios de los estados. Esta es una de las ventajas que ya comentó el presidente de Fundación UNAM al inicio, de poder alcanzar una audiencia mucho más grande que la que normalmente teníamos en forma presencial. Entonces, sí, es un reto enorme y está en nosotros ahora el aprovechar al máximo estas capacidades de comunicación y de informática. Aprovecho también para hacer una invitación de parte de Fundación UNAM, en donde bueno, nos acompañan el presidente de Fundación, licenciado Dionisio Meade, y la directora ejecutiva, la licenciada Araceli Rodríguez de Fernández, que tenemos muchas otras actividades en marcha, entre ellas la del Foro 2020, que ha sido uno de los programas más exitosos en Fundación, y que el próximo 11 de agosto tenemos el inicio del ciclo de lecciones de la pandemia, con una conferencia muy interesante y muy relevante a la situación en que nos encontramos. Entonces, pues mi agradecimiento a Mario, un saludo muy afectuoso para todos los que nos acompañan, y le paso con mucho gusto la palabra a nuestro presidente de Fundación UNAM, el licenciado Dionisio Meade.
Muchas gracias, Jaime, por tus palabras. Muchas gracias a Mario por esta presentación formidable, y muchas gracias por todo el ciclo. Yo creo que así como esta iniciativa de Universidades por la Ciencia ha significado eslabonarnos en distintas universidades del mundo y tener como en este momento participación de muchos interesados de distintas partes del planeta, hay que subrayar que otra de las lecciones que nos está dejando la pandemia es el renovado interés por conocer nuestro planeta, por conocer nuestra historia, por conocer nuestra evolución. Hoy son cada vez más los que se dan cuenta de la importancia que hay detrás de este conocimiento y de este cuidado por el conocimiento del planeta. De manera que celebramos que esto esté ocurriendo, y agradecemos también a Jaime la referencia que ha hecho a nuestro programa del Foro 2020. Efectivamente, lo tendremos también su inauguración el próximo martes 11 de abril a las 5 de la tarde, y ojalá podamos tener también esta concurrencia multidisciplinaria, multiuniversitaria y multipresencial de distintos lugares del mundo. La presentación será a cargo del doctor José Narro. Él hablará de las lecciones de la pandemia en México, y además de su experiencia como epidemiólogo, en su formación como médico, pues suma una larguísima trayectoria en el manejo de políticas públicas. Fue rector de nuestra universidad y seguramente aportará muchísimo en esta presentación, a la que de una vez ahora los estamos invitando. De manera que nos veremos el jueves, nos veremos el martes en la mañana, y los esperamos también el próximo martes en la tarde. Muchas gracias a todos. Un orgullo para Fundación UNAM participar en este evento. Gracias, Jaime. Gracias, Araxi. Gracias, Mario. Gracias a Martín, que espero por ahí esté, de la Universidad de las Américas, y que es también copatrocinador de este evento. Muchas gracias a todos y seguimos en contacto.
Muy bien, pues muchas gracias, y pues un saludo muy afectuoso a todos. Mario, y a todos los que nos han acompañado. Como siempre, es un gusto estar todos juntos y aprendiendo más sobre las diferentes investigaciones y acortando distancias, que es la intención que tiene este ciclo de Universidades por la Ciencia: acortar distancias y hacer sinergias. Muchísimas gracias.